Hi there. So last class we have seen about absorption of water and their mechanism. So today we are going to see about the translocation of solutes. So the translocation is the movement of organic food materials or the soluble solutes that are produced by the process called as photosynthesis, which will be transported from one place to the another, say from stem to root or root to stem in higher plants. So this whole mechanism is called as translocation. So now coming to the directions of translocation, there are three types of directions that translocation can happen. The first one is your downward translocation, the second one would be our upward translocation, and the third one would be the lateral translocation. In case of downward translocation, the organic material is manufactured in the leaves, as I said earlier, by the process called as photosynthesis, and it is translocated downward to the stem and the root parts. So it is then this organic food materials is then used for the consumption purpose, and it can also be used as a storage medium. And this translocation is facilitated by the phloem vessels, especially by the sieve tubes that are present in the phloem. Now regarding the upward translocation, this mainly takes place during the germination of seeds or tubers, or in the when the plant is in its seedling stage. So here the stored foods are being converted into soluble form and is applied to the upper growing part of the young seedling till it has developed the green leaves. As we all know, the photosynthesis needs chlorophyll, and the chlorophyll is present only in green leaves. So in the seedling stage the green leaves are still maturing. So here in that particular stage scenario, the upward translocation can be seen, and this translocation is also facilitated by phloem vessels, and under some special circumstances it can also happen through xylem vessels. The third one is a lateral translocation. In this type of translocation, the organic solutes will move from the pith to the cortex. So in cell we have certain anatomical parts called as pith and cortex. So in lateral translocation, a horizontal movement of solutes takes place from pith to cortex. These are especially seen in tree species, and this is facilitated by medullary rays. So, what are the evidences that say the phloem vessels are responsible for the translocation of organic solutes? So, the scientist named Malfigi in 1679 conducted an experiment known as the ringing experiment. So, in this experiment, he just cut a circle of bark from a tree. So, just exposing the xylem vessels but completely removing the phloem vessels just in between that wood. So after removing the bark, he could see the swelling of bark region just above where he made that cut. So in this swollen region, the organic solutes were getting accumulated as we have disrupted that continuous flow of organic solutes that was taking place through the phloem vessels. So this is a simple experiment that just proves that phloem vessels was indeed the responsible structure for the translocation process. Now, as the years went by, many more experiments were conducted. One such important experiment was done by Weidelf and Markle in the year 1944 using radioactive phosphorus, that is the P32. The radio labeled P32 was injected into the plant system, and a partition was made by using a wax layer to divide the xylem and phloem vessels. So this made sure that the P32 was kept to phloem vessels itself. So at the end of the experiment, the scientists recovered the radioactive P32 from phloem vessels and not from xylem vessels. So this is yet another experiment that proves that the phloem vessels are involved in the translocation process, and many such experiments were conducted by various scientists. So now coming to the mechanisms involved in the translocation of organic solutes, there have been three theories The first one is Munch's mass flow or the pressure flow hypothesis. The second one would be the diffusion hypothesis, and the third one would be the protoplasmic streaming hypothesis. And each of these hypotheses have its own positives and their drawbacks. So we will be seeing in a brief manner what are those features and what are those drawbacks. So coming to the pressure flow hypothesis. So this hypothesis compares the translocation process to that of the blood circulation system in humans. And in plants, the mesophyll cells are considered to be the heart which pumps the organic solutes or the synthesized food materials. So there are mainly two types of force that are being involved in this process, namely the pumping force or the osmotic potential and the hydrostatic pressure. So now coming into this diagram, so this is the source region and this is the sink region. So in the source there are mesophils that are present in the leaves, and in the sink region there are mesophils that are present in stem, roots, or whatever areas that are present other than that of leaves. So these mesophyll cells that are present in the leaves are the primary source for organic solutes, as the synthesis of food materials takes place in leaves via photosynthesis. So this is the source region where abundant of organic solutes are present. So thus the source with higher amount of solute obviously possesses a higher osmotic pressure. Hence these will absorb water from the adjacent xylem vessels. So this in turn will create a hydrostatic pressure. So these pressures force the solute through the sieve tubes. So this way these pressure will act upon the solutes and force the solutes through the sieve tubes. So these are the network of tubes which form a continuous chain within the plant body, which is helpful in the continuous flow of organic soluble solutes from leaves to other parts of the plant. So thus there is always a pressure gradient existing between the source and the sink, that is your mesophyll cells in the leaves and the mesophyll cells in the roots or stems. So also this pressure gradient remains consistently and remains throughout, as the leaves continuously produce food materials, and the osmotic pressure thus is always kept high, whereas the roots and stem would utilize these food materials for various metabolic activities, and the osmotic pressure or the potential here is always kept low. So such a pressure gradient is responsible for the mass or the bulk flow of these organic solutes. So now there are some major objections to this theory. So the first one would be that this theory explains the unidirectional flow of solutes. But whereas in plants, the flow of food materials or the direction of translocation is tend to be bidirectional at the given time. And the second objection to this theory is that it explains the translocation as a non-physiological process, whereas it is believed that translocation is not only a non-physiological process but also a physiological process. And the third objection is that the flow of solutes through the pores of the sieve tubes or the sieve plates are attributed to the excessive turgor pressure, which is also a major objection to this theory. So in plants, the movement of water or food materials or whatever the movement is happening is considered to be rapid. But the another objection to this theory is that the cross walls that are present in the sieve tubes offer a considerable amount of resistance, which in turn will prevent the rapid mass flow of food materials within the phloem tissues. So as we all know, the translocation process must be a rapid process, but these cross walls that are present in the sieve tubes offer some resistance or friction, which seems to prevent the rapid mass flow hypothesis. The second hypothesis is a diffusion hypothesis. This is very simple hypothesis which is based on the principles of diffusion, where the solute particle will move from a higher concentrated area to a region of lower concentration. So there will be a concentration gradient that is existing between source and sink, and the diffusion process will take place, which will ensure that the solutes have been translocated from leaves to roots. But this hypothesis doesn't have any relevance at this point of time, as the diffusion is a slow process. As I told earlier, the translocation or absorption of water or the movement of water or food particles would be rapid. So as the diffusion is a slow process, it can't account for the rapidness of the translocation process. So yet another variant of diffusion hypothesis was given by Mason and Phillips in the year 1937, which they termed as activated diffusion hypothesis. They stated that the stationary cytoplasm by activated diffusion and protoplasm provides the resistance-free medium, where they considered the diffusing particles, the diffusing organic solutes, they considered these particles to be activated or be charged. So, but in reality, this is only a theoretical concept, but in reality, there was no such activation of particles. Hence, these two hypotheses are just rejected. So the last one would be the protoplasmic streaming hypothesis. So this hypothesis was first proposed by Debris in the year 1885, which was later on elaborated by Curtis and her associates in the year 1935. So according to this theory, the moving protoplasm carries the solutes, or it can be also called as the protoplasmic fluids. So this carries the solutes within the sieve elements, and also these fluids can move from one cell to another through a large protoplasmic connections across the sieve plates. Also, a circular movement of living protoplasm has been observed. So, such a circular movement, it can also be termed as cyclosis of protoplasm, has been observed in different plant cells such as Chara, Nitella, and also in the staminal hairs of Tradescantia. And it has also been observed that the granular substance tend to stream in living cells. So it is also another evidence for protoplasmic streaming hypothesis. Again, this principle of streaming provides a more rapidness than that of the diffusion hypothesis. So apart from these evidences, in the year 1967, the scientist Stein gave a much more important evidence uh, that he found the existence of transcellular cytoplasmic strands which is present between phloem cells. So he suggested that these solutes are moving from one place to another through these transcellular cytoplasmic strands. So such a movement is facilitated by the energy giving molecule ATP, which is produced by the mitochondria. Though an important hypothesis, these also have some major objections, as there has been no streaming observed in matured sieve elements, and also the rate of streaming cannot be set in synchronized with the rapid flow of these organic solutes. So the rapidness and the movement of solutes, that is not the streaming that is not happening in the matured sieve elements, are the major roadblocks for this protoplasmic streaming hypothesis. So that's all for this lecture regarding the translocation of solutes. So thank you for listening. Hope I have improved a bit more than the last lecture. So I will try to improve much more in the coming lectures. And the next narration would be probably on the topic of photosynthesis. So see you till then.